Hi, my name is Lukman and I'm gonna explain about the introduction for the alkalinity test. The alkalinity of water is a measure of its capacity to neutralize acids. The alkalinity of natural water is due primarily to the salts of weak acids, although weak or strong bases may also contribute. The carbonates represent the major form of alkalinity since they are formed in considerable amounts from the action of carbon dioxide upon basic materials in the soil.
So the objectives of the alkalinity test are to gain knowledge of the methods used to measure the pH and alkalinity. Second is to be able to calibrate the pH meter. And lastly, it is to determine the alkalinity of a water sample and express it in terms of parts per million of calcium carbonate.
Assalamualaikum. My name is Irfan Zafri. I'm going to present about the concept of the alkalinity. The alkalinity of water is a measure of its capacity to neutralize acids. The alkalinity of natural waters is due to the salts of weak acids, although weak or strong bases may also contribute. Bicarbonates represent the major form of alkalinity since they are formed in considerable amounts from the action of carbon dioxide upon basic materials in the soil. Under certain conditions, natural waters may contain appreciable amounts of carbonate and hydroxide alkalinity. This condition is particularly true in surface water where algae are flourishing. The algae remove free and combine carbon dioxide from the water to such extent that pH values of 9 to 10 are often obtained. Boiler waters always contain carbonate and hydroxide alkalinity. Chemically treated waters, particularly those produced in lime or lime soda ash softening of water, contain carbonate and excess hydroxide and have further high pH values. This raises some objections from the consumers. For these reasons, standards are sometimes established on chemically treated waters.
Okay, now we move on to the procedure of alkalinity test. So this is the beaker, measuring cylinder, pH meter, magnetic stirrer, hot plate stirrer, hydrochloric acid and distilled water. First, we rinse the pH meter and 150 ml beaker with distilled water. Transfer 100 ml of water sample into the beaker and dip the pH electrode into the sample. Next, stir the sample with magnetic stirrer. Be sure that the tip of the pH electrode is near to the bottom of the beaker but not touch the magnetic stirrer. Record pH when constant reading is obtained. Add hydrochloric acid using burette in increments of 0.2 to 0.5 ml into the beaker. After each addition, mix the solution thoroughly but gently with the magnetic stirrer and avoid splashing. Again, record the pH when constant reading is obtained. Continue adding the titrant and measure pH until pH 4.0 is reached. Construct a titration curve by plotting observed pH values versus cumulative millimeters titrant added. From the curve, obtain the volumes of HCl added when pH is 8.3 and pH is 4.5. Compute the alkalinity values for both volumes of HCl in ppm of CaCO3. Hi, my name is Nurul Shahira. Now I will introduce about the BOD test. BOD, Biochemical Oxygen Demand Test, is a bioassay procedure that measures the oxygen consumed by bacteria from the decomposition of organic matter. The change in DO concentration is measured over a given period of time in water samples at a specified temperature.
The objective of this experiment is to determine the biochemical oxygen demand, which is BOD, for the water sample.
So to understand the concept of the BOD test, we must first know what BOD is. So BOD is usually defined as the amount of oxygen required by bacteria while stabilizing decomposable organic matter under aerobic conditions. The term decomposable may be interpreted as meaning that the organic matter can serve as food for the bacteria and energy is derived from its oxidation. In aerobic processes, oxygen is present, So heterotrophic bacteria will oxidize about a third of the colloidal and dissolved organic matter to stable end products, such as carbon dioxide and water, and convert the remaining 2/3 into new microbial cells that can be removed from the wastewater by settling. The overall biological conversion proceeds sequentially, with oxidation of carbonaceous material as the first step known as carbonaceous oxygen demand. Under continuing aerobic conditions, autotrophic bacteria will then convert the nitrogen in organic compounds to nitrates, which is known as the nitrification oxygen demand.
Now, we will proceed to the procedure of the experiment. So, I'll prepare 1 liter of the distilled water, and then I will add 1 milliliter of the magnesium sulfate. And then I will add 1 milliliter of the ferric chloride.
Hab die Welt verflucht, in den Wind gespuckt und salziges Wasser.
1 milliliter of the phosphate buffer. One milliliter of the calcium chloride.
War ziemliche Ruhe und die Sonne stand senkrecht am Himmel. Als ich über die Reling sah, da glaubte ich zu träumen. Da waren 1000 Boote und sie hielten auf uns zu.
I will still the solution. Dissolve the solution. And then I will pour the solution into the BOD nutrient barrier.
Sie sangen ein Lied, das kam seltsam bekannt vor. Aber so hab ich's noch nie gehört.
Switch on the bar.
So hab ich's noch nie.
And then it will aerate the solution about 30 minutes. Next, this is the water sample. 100 milliliter water sample, and then this is the water from the BOD nutrient barrier. I will add the 100 milliliter into the here, and then I will put it on the hot flat stirrer, burn it, and then I will put this inside, and then the pH will be 6.5 to 7.5. This is the solution. After that, I will put on the hot flat stirrer, and then I will put the DO electrode inside, and then we will read the benchtop Joule meter. After that, we pour the water inside the BOD bottle until full and ensure there is no bubble inside. And last, place the measured DO for the BOD bottle inside the incubator for 5 days.
Total suspended solids, or better known as TSS, refers to the dry weight of suspended particles which are not dissolved in a sample of water. TSS can be trapped by a filter that is analyzed by using a filtration apparatus. It is also a water quality parameter that is used to assess the quality of a specimen of any type of water, water body, or even wastewater treatment in water treatment plant.
Hello, my name is Irfan Zafri. So the objective of this experiment is to determine the concentration of total suspended solid, which is TSS, in the water sample.
Now, I will proceed to the concept of the total suspended solid. Total suspended solid includes all particles which not pass through the filter. Suspended solids are present in sanitary wastewater and also the industrial wastewater. There are also non-point sources of the wastewater, such as wastewater from the agricultural activities. As suspended solid increases, a water body begins to face its ability to sustain an aquatic life. Suspended solid absorb the heat from the sunlight and increases the water temperature, however, subsequently decreases the level of the dissolved oxygen. Photosynthesis decreases since less sunlight. First part of the procedure is to prepare the fiber filter disc. The filter disc is then washed successively 3 times using distilled water. Then the filter disc is placed in aluminum dish and dried in oven, which then it will be weighed. Next, a sample which will not yield more than 200 mg total suspended solid is selected. The sample is shaken vigorously, then the sample was passed through the filter disc. Finally, the filter disc is then dried in the oven and then weighed to acquire the total suspended solid of the sample.